Hi, everyone. We are going to talk about chapter 3 today. Chapter 3 is about cell structure and function. If you've ever taken a biology class at any point in your life, you always talk about cells and how they function. It's one of the first things we talk about. But what is a cell? We're going to talk about a few definitions of a cell first. So the cell theory, we talked about what a theory was and how a theory has lots and lots of evidence behind it when it becomes an actual theory. But there are three principles to the cell theory. A cell is the basic unit of life. All living things are made up of cells, and new cells arise only from pre-existing cells. Once upon a time, obviously, the first cells arose somehow. That is more for an evolution class when we get into it. Here we just start with new cells arise from pre-existing cells. Cells are small because of their surface area to volume ratio. Smaller cells have a larger amount of surface area compared to the volume, but an increase in surface area allows for more nutrients to pass into the cell and more wastes to exit the cell. There's a limit to how large a cell can be while remaining efficient and metabolically active. I'm going to link this video into a note. It's a good video to watch about cell surface to volume ratio. Once they get a certain size, then that's when cell division happens and they split.
In this video,
Microscopy. We in classes use a compound light microscope in general classes. There are way more advanced microscopes, but those are for like graduate students who are doing their theses in something having to do with an organism under the microscope. But this one has lower magnification than other microscopes. It uses glass lenses and light beams to view images. It can view live specimens. In classes, biology classes a lot of the time, a popular lab is to take a drop of pond water, drop it in there and see what little creepy crawlies that we can see. There are also electron microscopes. These use a beam of electrons, which result in 100-fold better resolution than a light microscope, but you can't look at anything alive. Anything you look at under an electron microscope has to be dead. So there's a scanning electron microscope for scanning the external surface of things. That's a paramecium on the left and all of its little cilia all around it. And on the right, there's a picture of the same thing, actually, a paramecium under a transmission electron micrograph or microscope for viewing internal structures. So next section is about how cells are organized. There's a lot of stuff inside of a cell. So there's two main categories of cells. There's prokaryotic cells. We call them prokaryotes for short sometimes. They do not have a nucleus. They have a nucleoid region in the middle where all the DNA is kind of condensed, but the nucleus is not separated off by its own membrane. The prokaryotes include two groups of bacteria called eubacteria and archaebacteria. We talked about that a little bit when we talked about classification in the last chapter. And then eukaryotic cells have a nucleus, which is their DNA is actually separated by its own membrane, its own nuclear membrane. And these include animals, plants, fungi, and protists. Commonalities. Prokaryotes, eukaryotes all have some similar commonalities. They're all bound by a thin plasma membrane. Inside all cells is a thick jelly-like fluid called the cytoplasm, where the cellular components are suspended. They're stuck in there like a Jell-O salad. All cells have one or more chromosomes carrying genes made of DNA, and all cells have ribosomes, which are tiny structures that build proteins according to the instructions from the DNA. We will get into more detail about all of this. But all cells, prokaryotic and eukaryotic, have at least these four things in common. So eukaryotic cells have organelles, they're mini organs, membrane enclosed structures that perform specific functions. They're embedded into the cytoplasm. Like I said, it has the consistency of Jell-O, the cytoplasm, where fruit salad is kind of stuck in there, and it can wiggle around a little bit because it's semi-solid, but that's what the cytoplasm is like. The first cells on Earth were prokaryotes, the archaeans. The atmosphere, way back billions of years ago, there was no oxygen. So some archaeans still survive in very hospitable conditions today. Volcanoes, thermal vents, salt lakes. That's where the archaeans live. Eukaryotic cells evolved from the archaea by a process called endosymbiosis. organelles may have developed from eukaryotes that would engulf prokaryotic cells. There's kind of a little gif of it there, but again, this topic is more for an evolution class. So cells gain a nucleus by the plasma membrane invaginating, which just means making a little pocket surrounding the DNA with their double membrane. The nucleus allows specific functions to be assigned, freeing up cellular resources for other work. Cells gain an endomembrane system by proliferation of a membrane. The increased surface area allows the higher rate of transport of materials within cells. Cells gain a mitochondria. Ability to metabolize sugar in the presence of oxygen enables greater function and success. So as oxygen came into our atmosphere, organisms had to figure out a way to process it. And then cells gained chloroplasts. The ability to produce sugars from sunlight enabled greater function and success for that. So that's just a short history of a few of the organelles, how eukaryotic cells got their membrane and mitochondria and chloroplasts. The next section is strictly about the plasma membrane and how substances cross it. So the structure is a phospholipid bilayer with proteins that are attached and embedded. When phospholipids are placed in water, they naturally form a spherical bilayer. Sorry. On the left, that is one phospholipid. It is polar, it likes water, so it will arrange, the heads will arrange itself on the outside and on the inside of the cell where water is. And then it has two little tails. Those two little tails are hydrophobic. It does not like water, so they are attracted to each other, and it's this yellow layer on the inside of that, the inside of the two layers of heads. This is called the fluid mosaic model. Scientists use this model to describe the membrane. Fluid just means it's flexible, the layers move against each other. And then mosaic, a scientist once upon a time thought it looked like a work of art, so different molecules make a pattern. There's proteins stuck in the plasma membranes in between all of the phospholipids. This is for transport, to move materials in and out. There's carbohydrates. Carbohydrates are like the ID tags to tell different cells apart. Our brain cells have different ID tags than our stomach cells, than our liver cells, our heart cells. And then there's cholesterol that's weaved through like yarn, makes the membrane stronger. Selective permeability is a characteristic that we attribute to the cell membrane. We also use the word semi-permeable. Semi means part or half, and then permeable refers to what can or cannot pass through the membrane. The cell membrane is said to be semi-permeable because it only lets certain molecules in or out of the cell. Generally, it has to do with size. Small things can float across fairly easily. Bigger things need those proteins to open and close doorways. Membrane proteins. Cells must control the flow of materials to and from their environment. They do this via the many proteins that are embedded within their membranes. Transport proteins are located within membranes and help move substances across. You see a little GIF where that is happening. Just the general idea of there's your protein embedded into the cell membrane, and then there's materials that are crossing it. It's too big just to float across. It needs a doorway that's a little bit bigger. So functions, transport. There's passive transport, diffusion, osmosis, and facilitated diffusion. There are three types of passive transport. Passive means does not use energy. And then there's active transport, where the cell has to use energy to actively pump things across. Exocytosis and endocytosis are two processes that are considered active transport. So, like I said, passive transport, the movement of a substance across the membrane without the input of energy. Diffusion is the random movement of molecules from a higher concentration to a lower concentration until they are equally distributed. This is a lot with smell. If somebody's in a corner of the room and they spray perfume all over themselves, the perfume molecules start really concentrated around that person, but eventually those molecules will diffuse and spread out across the room until there is an even distribution. Molecules move in both directions, but the net movement is from high to low concentration. At equilibrium, we have the same number of molecules move in and out of this cell, because when we talk about transport and diffusion, we're usually talking about what's crossing the cell and how much is on one side versus the other. During passive transport, we say substances diffuse down their concentration gradient. So the concentration gradient is the gradual difference in concentration of a dissolved substance in a solution between a region of high density and one of lower density. The molecules start off with the high density and then they diffuse into the lower density until it's all even. Osmosis is the special word for the diffusion of water molecules from high to low water concentration, and something called osmotic pressure drives osmosis. There's a pressure for those water molecules to spread out evenly on both sides of the membrane. Osmosis and water balance. So we use these three words, hypertonic, hypotonic, and isotonic, when we talk about solutions. And I like to liken it to Kool-Aid because it's a little easier to understand. But a hypertonic, you know what a hyper child is? They have too much energy. So a hypertonic solution has a higher concentration of solute. In a Kool-Aid solution, that would mean way too much sugar and not enough water. A hypotonic solution is the opposite of hyper, has a lower concentration of solute, where if you drink a glass of Kool-Aid and there's not enough sugar and too much water, you would call that a hypotonic solution. And then an isotonic solution has an equal concentration of solute. And in Kool-Aid terms, we would say that's just right, just sweet enough. Tonicity is the ability of an extracellular solution to make water move into or out of a body cell by osmosis. Normally, our extracellular body fluids are isotonic to our cells. Hypotonic solutions have fewer solutes. In this case, our blood cells will swell and can burst. Lysis is the fancy science word for bursting. And then a hypertonic solution have more solutes and the cells will shrink. This happens a lot with salt. Salt sucks. Salt will suck water out of anything you put it in, anything you put it on. If you put salt on a snail, the snail dies because it sucks all the water out of that snail. Osmoregulation is the control of water balance within a cell or organism, and we have a whole system inside that does this for us. Our urinary system is designed to keep our water balanced. And then you can see the three solutions. The isotonic solution, everybody's happy. A hypotonic solution, the interstitial fluid is less concentrated than the cytosol, than the cytoplasm inside the cell, so the water will swell, or the cell will swell with water. And then the hypertonic solution, the interstitial fluid is more concentration than the cytosol, so it will pull water out of the cell, and that's when the cell lyses. Facilitated transport. Some substances do not cross membranes spontaneously, or they may cross very slowly. These substances can be transported via facilitated diffusion. Specific transport proteins just act as open doorways. There's no energy input is needed. The molecules that are small enough just float across the membrane via the open doorway. Facilitate just means to help. So some of these bigger molecules just need a little extra help getting across the membrane. So some of our proteins are just open. They just open a bigger doorway and the molecules just float through. Still doesn't use energy, still a form of passive transport. Now we get to active transport as the movement of molecules from a lower to a higher concentration. So we are going against the concentration gradient. We use ATP as energy. We talked about that a little bit last chapter. It requires a protein carrier, which is often called a pump. For example, the sodium-potassium pump is active in all of our cells, moving sodium ions out and potassium ions in. Cells use bulk transport to move large molecules across the membrane. Endocytosis transports molecules or cells into the cell via that invagination process of the plasma membrane to form a vesicle. Phagocytosis is the endocytosis of pathogens like bacteria by white blood cells. Penocytosis is the endocytosis of fluid with small particles. And then receptor-mediated endocytosis. Particles first bind to receptors in the plasma membrane, which initiates that process of endocytosis. Exocytosis, endo means in, exo means out. These transports molecules outside of the cell via the fusion of a vesicle with the plasma membrane, and then it just kind of spits it out. We'll see a GIF in a minute that will make more sense. This is how signaling molecules called neurotransmitters leave one nerve cell to excite the next nerve cell or a muscle cell. Receptor-mediated exocytosis, uses a receptor on the surface of the cell to concentrate specific molecules of interest. So on the right, that is exocytosis. The vesicle just fuses with the membrane and kind of spits out whatever that is. And on the left, you have endocytosis. Wait till it starts over. That purple molecule needs to get in the cell, so the cell membrane engulfs it and then pulls it in. The nucleus in the endomembrane system is next. So the nucleus contains genetic instructions for manufacturing the proteins that are involved in most cellular functions. The endomembrane system is a series of membranous organelles that function to process materials for the cell. And there's the whole system there in the picture. We will talk about them individually here. The nucleus contains DNA in the form of chromatin most of the time, and then chromosomes when the cell is dividing. So it's chromatin when it just looks like loose strings. And then when cell division is upon us, it thickens up and it actually forms kind of an X shaped structure. We'll talk about that when we get to cell division at the end of the book. But the nucleus is separated from the cytoplasm by a double membrane called the nuclear envelope. There are pores in the envelope that allow materials to move between the nucleus and the cytoplasm. The nucleus has its own plasm. We call it nucleoplasm. It's the fluid inside the nucleus that's akin to the cytoplasm inside the whole cell. And then it has a nucleolus, which is the very dark region inside the nucleus that produces ribosomes, or pieces of ribosomes to be put together during protein synthesis. So there's all your nuclear structures. So the nucleus is the entire large structure, and then inside the nucleus you have the little darker structure called the nucleolus. Ribosomes are made of rRNA and protein. They are the sites of protein synthesis. This is where proteins are actually made. They can be found attached to the endoplasmic reticulum or free-floating in the cytoplasm. Both are shown in the picture. When free-floating, they occur singly or in groups called polyribosomes. Protein synthesized ribosomes attached to the endoplasmic reticulum, they have a different destination from those made on the free-floating ribosomes. So the whole system consists of the nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosomes, and vesicles. These are all different organelles found inside of every single one of a eukaryotic cell. We are made of billions of eukaryotic cells, and every single one has all of these parts. This functions to compartmentalize the cell and transport substances throughout. The endoplasmic reticulum is a mouthful, so we usually end up just calling it ER. It's one of the main manufacturing facilities in a cell. It produces an enormous variety of molecules. It's connected to the nuclear envelope, is composed of smooth and rough ER. And it just refers to the way it looks. The rough ER refers to the ribosomes that stud the outside of the portion of the ER membrane. These ribosomes produce membrane proteins and secretory proteins. Some products manufactured by the rough ER are dispatched to another location by transport vesicles, which are sacs made of the membrane that bud off from the rough ER. So you can see on the right the bottom right is a little GIF. The little bubbles that are budding off of it, those are transport vesicles. The rough ER lack the smooth ER. That's supposed to say smooth. We're gonna do it. The smooth ER lacks ribosomes, so it looks smooth. It produces lipids, including steroids. It helps liver cells detoxify circulating drugs. And it is attached to the rough ER, as you can see in the picture. You have the nucleus in the middle. Attached to that is the rough ER with all the ribosomes. And then on the side is the smooth ER that does not have the ribosomes. The Golgi apparatus, which can also be called the Golgi complex or the Golgi body. I don't know why we have so many names for it, but they all mean the same thing. It receives the proteins from the ER to repackage, store, and ship out to the rest of the cell. And for that reason, it is known as the post office or the UPS or the FedEx of the cell. Lysosomes are specialized vesicles made by the Golgi that contain hydrolytic enzymes, which break down molecules into smaller parts. They are prevalent in white blood cells that will engulf disease-causing microbes so they can destroy them. They break down damaged organelles. Our cells die every once in a while, and our organelles get tired inside. They need to be destroyed and recycled. They also sculpt tissues during embryonic development, helping to form structures such as fingers. We are webbed when we are in the womb, but obviously, our webs, for most of us, go away. And it's because of those hydrolytic enzymes inside the lysosomes that will destroy the webs in between our fingers before we are born. The cytoskeleton, cell movement, and cell junctions is next. So the cytoskeleton does the exact same thing that our actual skeleton does. It maintains the cell shape, it anchors and moves the organelles in the cell. It's what our skeleton does for us. It gives us shape and it anchors all of our organs. It can be made of 3 types of fibers. There's microtubules, which are the largest. The middle size are called intermediate filaments, and there are thin actin filaments that make up the cytoskeleton. Microtubule assembly is controlled by the centrosome. It helps maintain cell shape since they are the largest ones. It acts as tracks along which organelles move, and then during cell division, it forms the spindle apparatus, which helps move the chromosomes. Well, that picture on the right, the green portion, those are the microtubules forming the spindle apparatus. In the middle, all of that blue are chromosomes, and the spindle apparatus will pull those chromosomes apart during cell division. Actin filaments, made of the protein actin, they're long and very thin, they involve movement. And then intermediate filaments are sized in between the two, they're intermediately sized, and their functions vary. Cilia and flagella are the 2 movement structures that help cells move. Both are made of microtubules. Both are used in movement of materials along the plasma membrane or cellular movement. Cilia in the respiratory tract move mucus towards our throat and our nose, and then their flagella on a sperm propels them toward the egg. Animal cells secrete a sticky glue-like substance called the extracellular matrix that holds tissues together. The surfaces of most animal cells contain cell junctions, which are structures that connect cells together into tissues and allow them to function in a coordinated way. Our heart is an organ that needs to function in a coordinated way. Every single cell has to beat together, or else our heart is not working properly. But that is also a great video about cell-to-cell osseointegration. I will put that in a note for you. The extracellular matrix, a protective mesh of proteins and polysaccharides, surrounds the cell that produces it. It contains collagen, which resists stretching, and elastin, which provides resilience. Fibronectin is an adhesive protein that binds to integrin, which is an integral membrane protein, and that is connected to the cytoskeleton. And this all plays a role in cell signaling. Our cells all talk to each other. Cell junctions, that just means gaps. There are gaps in between our cells. There's different kinds of gaps. We call them junctions. Adhesion junctions attach to cytoskeletons of adjacent cells. So our cells will stick together and they won't move all willy-nilly. Tight junctions produce a barrier, and then gap junctions are when 2 channels fuse, allowing communication between the cells. There's examples of each on the bottom. Metabolism and the energy reaction. Metabolism, all chemical reactions that occur in the body. These occur via various pathways. They begin with a particular reactant and end with a final product. Many steps can be involved. Each is a chemical reaction catalyzed by an enzyme. The substrates for the first reaction are converted into products, and those products become the substrates for the next reaction. That's why it's a pathway. This makes this, now this makes this, now this makes this. So one reaction leads to the next in an organized and highly regulated manner. Feedback inhibition is when the end product interacts with an enzyme early on, a regulation to slow the pathway so the cell does not produce more product than it needs. So in this case, whatever D is the end product, once we make enough of it, it will inhibit that first step. Say, we have enough, now you can stop. Enzymes are protein molecules that speed up the rate of chemical reactions inside living things. They are often named for the molecules that they work on, called substrates. So lipids are broken down by lipase. Sucrose, the sugar, is broken down by sucrase, the enzyme. Enzymes normally end in ASE. All living cells contain thousands of different enzymes, each promoting a different chemical reaction. They are not used up in a reaction. Our body reuses enzymes all the time. It will break down the substrate or build up two substrates into one and then just get reused and keep performing the same job. So this is an example of an enzyme reaction. Number one, ready for substrate. The enzyme is the big fat purple thing. The place on the top where the substrate will fit in is called the active site. The substrate will come in this case, the substrate is sucrose in this example. So number two over here, the substrate and the enzyme bind in that active site. Water will come in, perform hydrolysis, and split the sucrose into two monosaccharides, glucose and fructose, and the product is released. Enzymes work by lowering the activation energy of a chemical reaction. Activation energy is the energy that is needed to start a chemical reaction. It's kind of like coffee in the morning. Those of you that drink coffee needed to start our day. Those are our enzymes. Some enzymes are aided by non-protein molecules called coenzymes. Vitamins are often components of coenzymes. So here's a graph. Each of these lines represent some sort of chemical reaction. It doesn't really matter what the chemical reaction is. The blue line is a chemical reaction without an enzyme. So it's like pushing a boulder up a hill. It's a really, really high hill. It goes really, really slowly. And then all of a sudden, once you get to the top, then the reaction can start. On the bottom, when the enzyme is present, all it does is lower that hill a little bit. So you don't have to push the boulder up so far. So the activation energy is lowered in the chemical reaction with the enzyme. Molecules store varying amounts of potential energy in the arrangement of their atoms, specifically in their bonds. This chemical energy can be released when the bonds are broken during a chemical reaction. Organic compounds are relatively rich in this chemical energy. Like organic is a fancy word for made of carbon. All of our food is made of these long carbon chains. We eat it. Those bonds get broken, we get energy. It's actually a very simple process. Cellular respiration is one such chemical reaction. It takes place in the mitochondria. There's two parts to it: the chemical breakdown of the food molecules that we eat, and then the storage of that energy in a form the cell can use to perform work, which is an ATP molecule. So at the bottom, you have the actual chemical reaction in both words and symbols. Glucose plus oxygen. Bonds are broken, they rearrange. Products are carbon dioxide, water, and energy is also produced in the form of ATP. The structure of a mitochondria, now that we are talking about cellular respiration, mitochondria has an inner and outer membrane of its own. The inner membrane is folded into cristae that contain enzymes for cellular respiration reactions. They might have been prokaryotes long ago. They have their own DNA and they are able to reproduce. Our mitochondria reproduce themselves inside of our cells. The ATP-ADP cycle, ADP cycle. It's a repeated cycle of production of ATP in a cell. When energy is needed, ATP is broken down into ADP, adenosine diphosphate, and that single individual phosphate molecule. When energy is obtained from food, it is used to add a phosphate back onto ADP to make ATP. So we have that potential energy for later. So you can see the GIF is just this entire cycle of ATP production. There's three pathways involved in cellular respiration: glycolysis, the citric acid cycle, or the Krebs cycle, and the electron transport chain. These pathways allow the energy to be released slowly. A tremendous amount of heat would be lost if glucose breakdown occurred all at once. We go through these pretty lightly, these three. There are a lot of steps and a lot of details to these, but we just skim through the processes. So glycolysis, the breakdown of sugar. breaks glucose into two 3-carbon pyruvates. So glucose is a 6-carbon sugar. Just break it in half into 3. This occurs in the cytoplasm of almost every cell type. This process is anaerobic, means it does not require oxygen. And NAD and 2 ATP molecules are also produced. NADH are other energy molecules that our body uses besides ATP. So you can see it very simply in that GIF. You start with 6, they get split into 3. When oxygen is not available, fermentation occurs. When oxygen is available, the pyruvate then enters the prep reaction, which prepares the pyruvates for use in the citric acid cycle. This is also known as the Krebs cycle after the scientist who figured it out. It is a cyclical series of enzymatic reactions. It occurs in the matrix of the mitochondria. It completes the breakdown of glucose by breaking the bonds between carbons. Each pyruvate enters the citric acid cycle as acetyl-CoA, produces NADH and 2 ATP, and it releases, this is the step that releases carbon dioxide. So the remaining hydrogens and electrons are carried by NADH and a similar molecule, FADH2. Fats and proteins may be converted to compounds that can enter the citric acid cycle, but normally it's just sugar. As you can see, there's a lot of steps and a lot of detail to this. We are just skimming the surface. And then the final step is the electron transport chain. NADH from glycolysis and the citric acid cycle deliver electrons to the electron transport chain. The members of the electron transport chain are carrier proteins embedded in the mitochondria cristae. Each carrier accepts 2 electrons and then passes them on to the next carrier. Oxygen is the final acceptor of the electron at the end of the chain, so the chain is aerobic because oxygen is needed. After oxygen receives the electrons, it combines with hydrogens and becomes water. The reason why we breathe oxygen is so that it can receive electrons at the end of the electron transport chain. The energy released during cellular respiration is used to make 36 to 38 ATP molecules. And we do this with every single breath we take and with every single time our cells are constantly going through this entire process. Because our body needs energy for everything it does. Fermentation is an anaerobic process that does not require oxygen. So when oxygen is not available, it is not present to accept electrons, so the electron transport chain is inoperative. It doesn't get used. Glycolysis still usually occurs, but only 2 ATP molecules are produced per 1 glucose molecule, and it produces lactate, which is a toxic side effect. That is all for chapter 3.